we all should know, the young students, that the Department of Plant Science in the Central University of Kerala was established by Professor G.M. Nair, sir. I request all to recognize and we have to appreciate him. Because of you, sir, we are growing up and we are here by your initiation and insight for the department. Currently, sir is a director, honorary director of the Central Laboratory for Instrumentation and Facilitation, University of Kerala, a flagship establishment of the university. Apart from that, sir is regularly supporting for the Central University of Kerala as expert in various capacities. I request our such a eminent professor, we are very happy, and also I am happy that I, as a chair with you to share the platform of your this special lead plenary lecture.
In fact, I was also very happy that the current Vice Chancellor, Dr. Sidhu P. Algur, and me were together on the day he joined this place, So I had the pleasure to welcome him in one of the meetings organized by the biochemistry department. So I suggested to him that certain things that is needed. So he accepted. I don't know how far he would go in doing it, but then one thing that I told him is that there is no signature building, See, every Kerala University has got a signature building. And Institute of Science has a signature building. Just put the photo, people will understand which institution it is. But you don't have, because almost all of them look alike. And in the master plan, there was a provision to have a signature building, what you call as a kind of iconic building, which would reflect the culture of Kerala as well as to that of the strength of the university, Central University of Kerala. So I told him, and he said he would do it. I don't know. And he was also feeling bad because there is no Kerala architecture in many of the buildings, So he said that he expected many buildings to be made, Becker and Shankar's model, alligator kind of facility, because when you are in Kerala, so that would have been great. And I'm sure that you also should urge that there should be a signature building, that will come. Kerala University Tower will come in all the kinds of things. So like many other universities. And so I think that's one thing. So I was asked by Jasmine and Dr. Rajendra that research ecosystem in India with special reference to Kerala. Implications for improvement, startup enablement and funding process. It's a big topic as such. In fact, I was exclusively working on this to speak to a larger audience, but then I interpolated certain biotechnology, botany into it. So maybe that the first part of it, I will just go a little high speed. But you have listened to people today, especially to Dr. Ananda Ramakrishnan, who represents the CSIR NIST system. Everything that they do is always there is a eye of technology transfer. And most of the things that they do has a kind of relevance to the societal needs. So either it is waste management, whether it is for ethanol production, maybe that's not for the society. Ethanol might be refined to something else anyway. But many other things, the waste management, biomedical waste management, all those that is. So I felt so happy that a central institute is addressing the problems of the state of Kerala. Normally they can do on their own, projecting themselves to the national, international arena, but then they always address that. And that's the greatness of Dr. Ananda Ramakrishnan and the CSIR NIST. And what you have learned from his lecture is that how exactly you can make simple ideas, innovative ideas to be transformed into something which would be useful to the society. That's what my initial theme of my talk is. Now this is very important that everyone should realize the government of India is going to enact this in the parliament, scientific social responsibility. You have a responsibility because you're all beneficiaries of the public money. And if you're doing a MSc course or PhD or any kind of course, there is a responsibility that you have to give to the society. Mostly what will come is as a product, or maybe addressing the environmental issues, the biodiversity issues, the health issues. All this could be something which would, that's what your responsibility is. So I think this will be enacted because we have a corporate social responsibility which is enacted long back, and the companies are following that 2% of their profit has to go into the charitable societal activities. So this, once it enacted, I'm sure that you will be answerable, and anything that you do should somehow, or in the immediate run or in the long run, should go into the society as a remedial measure for addressing the problems, or as a product. Could be health-related products, or maybe enzymes, industrial enzymes, or maybe many things can be shaped up if you understand this properly. So you have to voluntarily contribute your knowledge and resources to the widest spectrum of stakeholders in the society in a spirit of service and conspicuous reciprocity. That it's a kind of reciprocal understanding between the society and you, that you do something which would culminate in a product or a process which would be beneficial to us. That's what the essence of, it's a big document. I was also associated, fortunately I was also associated with the framing of this document, so that's why I just wonder. This will be enacted soon. Science and technology linkage so as to make S&T ecosystem in the country vibrant, bridging science society, science, and society-science gaps. There are big gaps these days because you don't have realized that responsibility vested onto you. You do it, you casually do research for the sake of doing it, or sometimes you may have some innovative ideas that you would do a research which would lead to some product or process. But mostly, you don't put your mind, you don't put your innovation. This is well researched, reflected in this particular kind of thing. We all do basic research, isn't it? Even when you speak of biotechnology also does. Very few biotechnological interventions go to the society as a product or a process. So there also it's all, though you say biotechnology as an applied science or many science, but the only science which goes to the people is the agricultural science. They work always for the society. Probably they would. But then what happens is that we normally concentrate on basic research, where publications, patents, these are too important because, as you know, that teachers sitting over here, unless they have publications, they cannot just get their career advancement. For you to go in to get a new job, I'm sure that you need to have good publications. So it is necessary. But then it's not that you should only do public. If you go to CSIR NEST, I would say each of the student will have minimum. I have evaluated their thesis, or maybe they came for open defense. Minimum 10 to 12 publications in standard journals will be there, in spite of putting many to the technology transfer sector or the product level. How do they manage? You go to ISET Trivandrum, you would see that it's a BSc, BS, MS course together, isn't it? And over there, I was very happy to see that youngsters in the second semester of their BSc course doing great and publishing papers in Nature, and they always think of doing something beneficial to the society. So my urge to you, to each generation, is that we talk of Vikasit Bharat. It's not that, unless individually you reflect your ideas and innovations into product and process, I'm sure nothing will happen to the country. Vikasit Bharat, maybe I'm told that we would become a Vikasit Bharat, a developed nation, soon, because we have all the potential. Space, we are at the top. And many of the sectors that we are, if you look at it, that we are superior to many people. So we are almost a Vikasit Bharat. But then to make it much more precise, the Prime Minister has termed this as 2047 as the time frame for making a Vikasit Bharat. That's all 100 years of independence. But I'm sure that we will become, provided the younger generation would rise to the occasion, do it, and do things great by which you can make it. So what I'm saying is that most of the research that happens, if you, this star, this. What? It's okay. No, it's okay. Now, if you look at the value of in most of those research, either it is, you call it as applied or basic, it's dumped, isn't it? You know, thousands of theses are being defended every year in every university. Maybe in different subjects, but then I'm sure in science stream also several of them. What happens to them? Do you look back at the research which, I'm not blaming you, because that's what the necessity of the situation. You would make a PhD thesis by hook or by crook, and then maybe that as there is a requirement for making 1 or 2 publications, you make it, then dump it. That's what the real value of death is. If you, I'm sure that all these thousands of theses, if you just rightly mine them out and find out the value of it, I'm sure that there will be treasures, because your idea was to get a PhD. And once you go to your PhD, your idea, you don't think about scientific social responsibility and all, isn't it? So your idea is to get a job. So to get a job that you wouldn't refine what you have done, because many times that you forget what you have done for your PhD. You won't look back at what you have done. You feel that it is all inferior and probably with great difficulty you have managed to do it. But that should not be the case. If it is there, and even you talk of applied research, even many of the applied research, as I said, also becomes like that. They might make some patents. As you know that patents are very important, we need to have. But patents have no value unless it is translated into a product. So you'll get a patent and it will be sealed. Your ideas, innovations will be sealed for 20 years. But if nothing is being, after 20 years, who needs your technology? So what is needed is that if you patent it, try to translate it and then make it a product or a process which goes to the society. That will have greater value. Otherwise, patents have no value. Publications, at least people will be reading it. Your patent, nobody. The only thing is that when you look at the database of patents, if it is approved, that somebody will know that there is a patent like this. Patent search is there. But otherwise, that has no value. So what I say is that translational research and fostering bench to bedside research is what is needed. Everyone cannot do it. Basic research, because there was a okay. This is what is happening. Now, if I sit in many of the committees of NRF, SERB, DST and all, But where I find is that almost all the projects are coming on basic aspects only. There is no applied research, or there is no translational value or translational component in any of the projects. That's why most of them are towards basic research. I don't say that basic research is not needed, because that's the foundation. You cannot have an applied research unless you have basics. But you should not stop there. That's what I'm saying. So the project will come. Maybe most of the cases the project is sanctioned to the principal investigator. The person would do that, and you please tell me when I have to stop it, because you know me. So I won't mind. I won't feel bad if you can. When lack of dedicated sustained support for prototype development, scale-up studies, regulatory trials, and market readiness. This is something which is very difficult that is happening, and everything. What I'm saying is that there is absolutely no fund available, but that I'm going to say, translational research. If you do a basic research and you feel that potential is there, you submit a second proposal, probably you may not get it. That connect is not there. So when you specifically establish that, I would feel that way, that if a person is going for the second time to an ANRF or a CRB project with translational component of the first one he has done, and that should be supported, but it doesn't happen that way. So that is one of those major constraints. That's what I say, that most of you all know it, isn't it? So when you speak of, many times they consider that since you have been given a project, no, don't come for the second one, So this is a kind of, but I try to do things in a different way. Shortage of professionals skilled in regulatory affairs, product design, scale-up engineering, and commercial and strategy. This is very important. Unless you have a knowledge of it, like traditional knowledge, the last talk was on traditional knowledge. Now, traditional knowledge is there. Some of them are codified and available in the public domain. But many of them are verbatim, word of mouth. People will just use it. There's no codified, there is no documented. They are not documented. And there is no, it's necessary if validation is necessary. If you feel that there is something that's valuable in it, probably you can validate it and then come to a product. The knowledge becomes useful only then. Otherwise, knowledge remains as a knowledge domain. Probably it would come in a database like TKDL, traditional knowledge database, or that kind of databases, but then nobody uses it. We were, at TBGRA, we were the first one to go in for that equitable benefit sharing process. On a tribal knowledge, we developed a drug called as Jeevani, and it was sold to a Ayurveda pharmacy, and half of it, what you call as equitable benefit sharing. That remains the first world and the last world model of equitable benefit. That's based on a knowledge of a tribe that we have given them, maintaining all the IPR things that we have given them. So knowledge systems are there, but we don't. Mostly what you do is that what your teacher says, your supervisor says. You would accept that research work and do it, isn't it? How much of innovation? I frankly ask you, how much of innovation that you do? How many of thought process that you do when you design a research problem? Tell me, confidently can you say? Teachers, I cannot blame them. They have a domain. See, you will be very comfortable if you come to that domain. And, if you, see, I had 30 PhDs have graduated with me for PhD. If you look at it, one or two, there may be repetition, but rest of them all, I left them. Dr. Suma did her MPhil, Dr. Jasmine did, and they know that most of and my students, even microbial technology, biodiversity, biotechnological aspects, tissue culture, single cell proteins, so many. So I leave it to them. But I'm so happy. I'm so happy that most of them are well placed because they could think independently, act, and become skillful. That's what is lacking. So if you are spoon-fed, I'm sure that it would be difficult for you. You ask Suma, you ask Jasmine, or many of those people, what was the kind of training that they got and then how they have refined themselves to take independently. And so that kind of vision should be. Innovation is the catchy word these days. You don't innovate. That's one of the major difficulties that you have. I'm sure that teachers would gladly accept it, provided you explain to them that this is the area I want to work. And how many of you dare to do that? Yeah, so if you do that, I'm sure you will be able. So translation from bench to market becomes inefficient and slow because of the reasons that we have. Poor market orientation. Most R&D projects are probably driven by science rather than by societal or market needs. When you innovate, when you do something, do you think about the society? You don't think. You never think about the society to frame a project work. You just frame a project work. And probably when you're in the comfort zone, you would love to do that, and you do not. So that's one of the major problems that you may lack practical viability or user adoption pathways. Fragmented research industry. This is very important. I'm sure this is what I'm advocating. When I was in the Kerala Biotechnology Commission, I have connected many industries to the research programs. One of the programs is partnering academic and industrial research, which we did, where an industry partner supports the student's research work. The student will work for the industry, and an institution is being aligned to the process. In the process, the student gets a very good fellowship. Student gets a PhD and also a placement back in the industry. And one of those very classical, my first pair program which we did, that girl joined an industry, and she is drawing more than, as you know, that when you join an industry, it's unimaginable kind of amount that you. And since she did that thing, it was just to develop a prototype, a handheld Raman spectroscope. It was a physics problem. But then she did it, made it in 3 years' time, published papers, and she was immediately, because the company got a product. She will also have the IPR rights of that, and it's a girl. And she became a role model of that. And certainly, subsequently, there were several ones. So industry should come forward, and I'm sure that after some time, maybe it would be very difficult to get fundings from the university or from. You need to have the industry, but for that you need to work for the industry relevant problems. They won't invest. They won't invest in anything unless they get a benefit out of it. So industries can be roped in, provided you will think about a problem, work for them, but there is absolutely nothing, because their product only will go there. You can make the publications. You don't have to go for a patent. Even if you patent it, then you can publish it. Once you publish it, you cannot patent it. Okay, but if you patent it, then you can publish. This is what it is. Because when you patent it, it should not be in the public domain. So a publication is in the public domain. So you patent it and then publish it. So absolutely no problem. This is what CSIR NIST does, or many other institutions does. So industrial partners are very important. This is again another. How many GLP, good laboratory practice or good manufacturing compliant labs we have? We don't have. Because when you speak of a product, you need to have certified laboratories to do it. There are a few laboratories over here. For example, TIMED in Sri Chitra. There are few institutions. Like the classical example of industry roped into the research system is IIT Chennai. Their innovation hub, I think there are thousands of startups, student startups, teacher startups, all of them are there. That's because, unless you have that kind of, so you need to have, I'm sure Central University should think of starting an incubation center, a technology transfer center, and all these are very much essential at the present time. I'm sure that the government sitting over there is not going to close their eyes, So maybe that they would block your grant or something of that unless you think of that way. So I don't know whether you have an incubation center. You need to have a common laboratory rather than making, fragmented facilities. You pull them into one place. I'm sure that when Dr. Venkateshwarlu was there, he came and visited CLIF, Central Laboratory for Instrumentation and Facilitation at University of Kerala. And he was very impressed, and he said he will immediately start it. I don't know what has happened to. Okay, if it comes, very good, because he had that vision to make it. Regulatory clearances is very important, because many times, that any product, any product that has to go to the human use should have the preclinical trials and clinical trials to be done. Preclinical trial is the one which is done onto the animals. So once you find their efficacy and analyze the toxicity, then only you go in for the human analysis. Now, this is one of the faculty. You always look at a comfort zone, isn't it? So I don't want to indulge. Why the hell I should take all these unwanted things? I'm comfortable in my laboratory doing my research. My teachers are there to support, and even the teachers feel that my students are there to support me. So maybe that this is a kind of mutual understanding that you don't think of. So, there are a lot of risk. Obviously, a startup, I'll just give you a classical example of Biocon. Biocon started by a lady called as Kiran Mazumdar. She is a fermentation technologist. 25 years back, no one believed her. She took this papain, enzyme, that was her first product, invested 25,000 rupees. Like Steve Jobs, she started in her garage. Garage is small. Now tell you, Biocon is the topmost industry in the world. Several $1 million industry. So could happen. Things would happen, provided that you have that determination. Otherwise, it's not a comfort zone. But once you are successful, there is no looking back. You just ask Kiran Mazumdar of the kind of hardships she has taken it. Engineering, medicine, many things being said and done. Nobody wants to, for example, Ayurveda. It's a wisdom that we have, isn't it? But you ask any allopathic person, he will not have faith in that. He said that's a humbug science. They wouldn't approve. So that means allopathic doctors don't believe in Ayurveda, and you have no interface between those two. Integrative medicine has come that way, but still people shy away from doing it. So I'm sure that many of your research problem can be done and translated well, provided that you have a medical professional over there. And that doesn't happen, because you cannot do a clinical trial unless you have a medical personnel with you. Many of the zoology or biochemistry people would understand it. So that's very important. Products often fail to meet world usable afford. So this has been the interface. We talk the FIUG program 2020, NEP 2020 speaks about multidisciplinary, holistic, and inclusive education, isn't it? Inclusive in the sense everyone sitting together and learning. Holistic means you learn everything. That's what the greatness of that science. I don't know how far we take it. And then multidisciplinary. You should think of multidisciplinary approach, because everyone should be component of your program, then only you will be able to do it. Now, it is what is to be done? The GDP. See that 0.7% of GDP is coming to the research. It's a neglected area. Neglected areas contrasted to 1.52% GDP. If it is increased to that, US 2.8% and China 2%, they're doing great. Their publications are great. Their research arena, they stand, China, US first, China second, and that way, that is being maintained over the years. That's mainly because of their investment is much more. Because unless you invest, you don't get it. Unless you are supported, you won't use your money, your salary to do a research, isn't it? Industries can, of course. Otherwise, you need to have support. So for that, unless the GDP investment increases, you will not be able to do. And harmonized regulatory guidelines are needed. Private sector investors, as I have already told you, 40% compared to 70% in advanced economies. At many places, it is that way. Triple help for R&D centers in academic institutions. Will you dare to do that? CUK, can you house an industry inside this? We have a give and take policy working together. So you will align your research problem as per the requirement of the industry. Industry will support you. What a great blessing it would be. But I don't think that anyone would venture to that. So you should bring them over here, because they have great strength. Let them build their infrastructure in the university campus. They would be very happy, because they would make all the instrumentations available. Only your human resources are being utilized. And probably the product, there will be a great revenue generation to the university, and then things would happen in a big way. So there are a few examples where the government has gone into, as I've said, Translational Research and Innovation Center, which is started in 3 places in the state by the state government. PAIR program, as I said already. PAIR partnership of accelerated innovation and research. Include industry partner program, DST and NRF has got this, the hub and spoke models. That you will have one major institution and there will be several spokes in it. Together they work, and I'm sure wonders would happen. This PAIR program is already in force, by NRF, and where, what you call it, the hub and the spoke. So you will have one institution which is ranked within 20 of NIRF ranking. That would be the hub, and then all the institutions. So anyone can join that, and probably the amount that is given is great. I'm sure that you don't have to just be in the crowd and get money for making it. So this PAIR program is going to, that PAIR acronym is borrowed from us. We started, Kerala State Council for Science and Technology started PAIR program some 10 years back. NRF started it only last year. But the acronym is PAIR. But there's no copyright for it, so anybody can claim it. And into IP, as I was telling, that IP is very important. How many of you are aware of the IP rights? If you make a product, maybe that there will be co-inventors. Your guide will be an inventor, you will be an inventor, an industrial partner will be an inventor. What would be the share? When you make a profit, and when you make the profit only, the problem comes. So initially you need to have IPR facility over here, understand that what this intellectual property rights concerning each and every. national infrastructure networks. So if you, for example, I represent CLIF, Central Laboratory for Instrumentation, many of you know it. Stick models in CUSAT. ISTEM. ISTEM is a connectivity established by Institute of Science with all the, instrumentation facilities across the country. So you need not write to CLIF now or to ISTEM. You just go to the ISTEM portal, register, and then ask for it. Wherever there is a slot available across the country, there are about, I think, about 2,000, 3,000 central instrumentation facilities are listed in the ISTEM, it is being called. Just put it, and then you will be directed to their website, and you can book anything. So what I'm saying is that absolutely no problem. You have everything with you. It's only that your vision, your mindset, and your commitment that is important. All the facilities are available. You want a cryo-EM, it is available. You want a HRTM, it is available. You want to have a mass spec, it is available. Everything. May not be here, but anywhere in the country, and it will be open to you. So make use of that ISTEM facility, which is very important. Skill Vigyan program, again another major program where skilling at different levels, technician levels, teachers levels, student levels, and there is a big program going on there. So that is what some apprehensions of the ecosystem development in Kerala. This is very important, not in Kerala alone, because we don't stand as such, but then obviously we need to think globally and act locally, because we need to look at our needs. So once you do that, then I will just close it. You know that bioprospecting is something which is very important, and I'm sure that anyone could take up this. Most of those great things have come from this. Not just because I was talking to, the last lecture which was talking about the Indian systems of knowledge, Indian knowledge systems. See, it's not that. There are so many novelty ideas available in the biological world. Do you know the colocasia lotus leaf effect? You know that you normally say lotus effect. Lotus effect is that when a drop of water falls in, it rolls down. It carries all the dirt along with it. You know, what kind of beautiful structure plant has devised that? And people have taken that know-how and now have ceramic tiles, put it in the bathroom, which will not hold a drop of water. It's done. So, and so many novelties are there available which could be used, biomimicking. So you could mimic that, and prospecting would involve that also. Structure. Have you seen Victoria regia? Victoria regia, the lotus which has got big leaves, they say that you can put a baby onto it. If you leave it, this only one stalk, and there is a ramified, labyrinthine structure over there, and that supports the leaf. Why can't you make a structure, architectural, a civil structure like that? People have done it. So if you see wherever in airports, if you see one stalk and then the large dome with no pillars are there, this is all know-how from plants only. So there are so much of, and you also, there's so much of, once you bioprospectors, this is a classical example that when you are a phytochemist, if you look at because plant kingdom contains thousands of molecules which could be refined into products. This lady, you see that? This lady, she was so young, she discovered that artemisinin, Artemisia annua, a plant, has got anti-malarial properties. We have been using cinchona, derived products because the parasite has developed resistance for it. So most of the anti-malarial drug does not work. She saved millions of people just by finding artemisinin, trying its efficacy, and then came out as a great product. So there's so much of plants, like what I showed you before. They're all plant products which are extensively used. They say that 10 maximum, prescribed medicines in the world are plant products. Not just chemical drugs, plant drugs. So there are umpteen options. Like Artemisinin, she got Nobel Prize in 2015. So you look at her now and look at this girl, young, beautiful lady. Like this is same thing happened to Barbara McClintock. She discovered jumping genes in 30 when he was in very young age. She got Nobel Prize when he was 85. So that time validation was not available. Now these days there are strict validation which are. Now, if I don't say this, you see, all those genetic engineering, absolutely genetic modifications do not happen these days, mainly because there are a group of people averse to it. They don't allow this kind of genetic modification. So GMOs are not getting into the farmers' field these days. But then we should owe to this great people who have, identified the restriction endonucleases, and then thereby the whole molecular genetics. Sanger, you know that. All those sequencing methods were done by. So Sanger, what is his uniqueness? He got Nobel Prize twice. He got Nobel Prize in 1958 for sequencing the insulin protein, and then for 1980 for nobody gets that way, because once you get a Nobel Prize, the money is so great that you forget everything, like what Carey Mullis did. So maybe that he got Nobel Prize twice. There are people, husband and wife, daughter sharing Nobel Prizes, like Marie Curie, father and son sharing the Nobel. But then very seldom that you get Nobel Prize twice in school. So he is great. And I'm sure that all those new generation technology, what you call, Carey Mullis, as I said, that, PCR is a revolution, isn't it? You could amplify DNA at nick of the time. And all the happenings, you could overcome the COVID menace or all those processes, mainly because of, the PCR being developed, because RT-PCR has become a classical term, which everyone knows, is mainly because of this. So, but then it's something very unique. It's a revolution that has happened. See, otherwise, it would have been great difficult for us to amplify the DNA. Now, within an hour's time, you can amplify any amount of DNA just from one piece. So, all this revolution is just mainly because of this man. He is no more. Now, all this sequencing has taken place, including the nanopore sequence, which have become very, very, very cheap these days. So maybe less than. Maybe that they say that 60, the instrument has become, the process has become very cheap these days. Next generation sequencing. So all those things, you can sequence your DNA within a nick of time. So that becomes, the revolution that has taken place in genomics is mainly because of this, what Carey Mullis and subsequent revolution in the sequencing technology done. Junk DNA, as you know that Andrew Fair and Craig Mello got it. It's many of those silencing processes, gene silencing, this was done. These are all revolutions that has happened. Carpenter and Jennifer, who have devised, CRISPR-Cas9 technology. And, they're not that old, so you could also, get something. But then this is again revolution. Because this is something, not genetic modification, but genetically modified organism. You don't have to borrow a gene from elsewhere. You can edit your genes and make it function differently. There are examples of that. And now, so I'll just quickly go to finish it on this. Synthetic biology, of course, it is making great, and we have a CVJ Center for Synthetic Biology in Cochin University being started on an industry-supported program. So synthetic biology has great role. You can devise anything that you want using that. Mean to say that I have to stop it? Okay, the gene editing. Of course, gene editing has been, this is a classical example. But have you heard of this? Onda. Sumaikku poണം. I wouldn't. Say, I'll finish it very fast. No, it doesn't look nice, But I cannot take that advantage with my student, Okay. this, I listened to this person about 2 months back. IARI has developed the first gene-edited rice varieties which can withstand abiotic stress. Just recent happening. Now imagine that there is no problem. Nobody should mudsling on this, because it is that its own DNA being modified by which it could resist the salinity and the temperature related issues. And if that plant is made that way, and if it has become a world model, just imagine. Activists again coming on to this is saying that there is no absolute, this is what is happening in the country. India, genetically modified organisms are everywhere. India, Kerala is a diabetic capital, isn't it? You know that. Kerala is, we are very proud that we are the diabetic capital. When you say about diabetic, you have to, many of them are insulin dependent people. Where does the insulin come from? It is through genetic modification, recombinant DNA technology. The one which is available as human insulin is made from the urine genes being expressed in bacteria, and humulin is being made. So the activists, if they are diabetic patients, they would not, just many times I ask them, can you stop taking insulin because it's a recombinant protein? They wouldn't, because they are all opportunistic, selfish. For them, they will use it, but they won't allow others to use it. That's what it is. So it's a question to be asked. I have been asking this to many people, but then this is what is happening. So everything, and they say activists are centered to withdraw genome editor, right? Before the technology has gone to the farmer's field, it is not even made to go to the farmer's field. They found something by which the plants could equip themselves to withstand that. No, where is the problem? There is no alien gene that has got it. You have not borrowed it from a bacterium. You have not transferred a gene from elsewhere. Its own DNA being done. But still, people do not believe in that. This is what is happening. The country is that. That's why there is a brain drain happening. The most convenient time you disappear from the country and go to Canada. What may be the job? Probably Dr. Jasmine did for Kerala Academy of Sciences. She did a survey that why students migrate to the other countries. There are several reasons for it. One thing is that you always ask questions. Why this, why that, why that? No questions asked. You go to Canada or US and you walk in a petrol bunk or in KFC or many other restaurants as a waiter, and you do your coursework over there. Nobody asks you. Probably what you would say that your supervisor also will be there working in a restaurant. Absolutely no. But here, see, if I'm a postgraduate and if I'm driving an auto rickshaw, the next thing is that all the media will be after him and to smash him. He will never take his auto rickshaw further. That's the kind of situation. This is the kind of situation. That's why we have been trying to bring them back. In spite of all the incentives given, they won't want to come back because of those obvious reasons. Organoid and tissue engineering, microfluidics. So this is very important. See, many of those who are doing animal experimentations, you need to have alternate models. So we have zebrafish, you have hydra, you have Drosophila. All these organisms are safe to use for animal experimentation. You cannot work on bigger animals, you cannot work on rat, mouse models, So why can't we mimic something? Organoid through synthetic biology is something, a miniature organ-like structure where you can do all the experiments. Now this is microfluidics, so this is something which is, just look at this, see, if you look at this, these are a microchip, and onto which you have micro wells over there. And each well will have cells from stem cells from different places being differentiated into heart cells, into kidney cells, into liver cells, and they are connected through the microfluid channels. So maybe that what you can do is that you just use a drug. And within the chip itself, it goes into the heart, goes into the kidney, and you would be able to understand the efficacy of the drug just by using. You don't have to use an animal model. So this is a wonderful technique that organ on a chip, human on a chip, these are all concepts that has come in, where you could do all these experiments. So this is what it is. So this is multiple organs. Each one it's a cells, being grown in the wells in a fluid situation. And all this would go organ on a chip, organs on a chip. So human on a chip, you can make it no questions asked, because you don't have to. The kind of ethical committees, human ethics committee is the most worst committee. They wouldn't allow anything to be done because you cannot experiment on human beings. Even animal ethics committee is much something. So probably you have to do away with animal experimentation. But you cannot get a drug approved unless you do animal. So how do you do that? So you need to find out alternate. Hydra models are being used. Drosophila models are being used. Zebrafish is being used. All these organisms are being used as animal models. They say that it's almost similar to the human beings. AI-driven biotechnology, I don't know how far this is going to revolutionize the biological concepts. Database generations, genomic database generations. That database creation, genomic data is plenty, but then to get value out of it is very important. So we have been using bioinformatic tools to find out, what the value is there, especially for the metagenomic process, where you pulled out the DNA and break it and then find out what are those genes, valuable genes, irrespective of the organisms. So for that, all these are needed. So maybe that there is so much of value in it, and I'm sure that being biologists, we had accepted information technology and computer science through bioinformatics and computational biology. Now I'm sure you need to go in for adopting AI technology. You should master it to get things done. Nanobiotechnology, as you know, the nano drug delivery, and it's again a big science that is happening. You can encapsulate your drug into a nanoparticle once it has got that ligands which would identify the receptors on the cancer cells. So the drug will go there and there will be slow releases that is happening. So don't you think it's a wonder that can happen? Delivering the drug to a site. Otherwise what is happening? You're consuming it, isn't it? So the whole body gets affected. So you could do that. Bioinformatics and computational biology, as I have said it. Bioprinting and 3D printing of tissues. Again, that bioprinting, like what Dr. Anand Ramakrishnan has shown about printing. That's printing, you could print anything that you want. You can print an organ. You can print anything because computer designs are available. You don't have to do anything. The printer would print the cells. I mean, on a gelatinous matrix, it would print a heart cell, heart shape into a heart cell, and the rest of things will be done. And once it is in a conducive environment, the vasculature will happen. So that's what is very important, because if a printed organ has to mimic a human organ, there has to be vasculature to be developed. So that again is possible provided the conducive environment. Angiogenesis, it's being called. Dr. Samir and his group is doing on that. Now, and this is just a classical example of how a lost ear is being regenerated through printing. It's not exaggeration. I think it has come in nature or something place where a printed ear is grown. So that means paving way that sometime in the course of time, regenerative medicine would work for recreating a lost limb, a ear, or any of the organ that is in your body. There is no tissue incompatibility being involved in it. So I don't know how far it would take to do perfect these technologies, but I'm sure instead of going after donors, and probably many of times it becomes difficult for you to spend that much money, the technology will become very, very cheap in the course of time. Agriculture and food technology. This is very important because you know that what has happened to the BT cotton, that is the only one, maybe before the activists became active, that this technology had gone to the farmer's field. So the only genetically modified crop that is available in the farmer's field is BT cotton. None of the other things have gone into the because this activists, so-called activists, have come against it. They have gone to the court, and there is a moratorium made on this, that you cannot use, or you cannot send that to a pharmaceutical. So all those genetic modifications rust in the scientist laboratory, and there is absolutely no translation that could be done. And especially to this brinjal, that BT brinjal was developed by Monsanto, Mexico, in India, but they were not allowed to use this technology, though they have generated so much of, spent a lot of money. But then what happened was Bangladesh got benefited. All the brinjal that is available in Bangladesh is genetically modified brinjal. There is absolutely nothing, see this one. This is the one, when you open it, you will see all this. But this is very, very great. And then you see this one, which was developed by Agriculture University, Tamil Nadu Agriculture University. So such technologies have gone to places, because why can't we accept it? Brinjal is always cooked and eaten, isn't it? So even if there is a gene or protein available, they get denatured through cooking. So only thing is something which is eaten raw, probably things. But I am sure brinjal, you don't eat brinjal raw, isn't it? So it could be. So, and GM mustard. So 10, 15 years, Deepak Pental of Delhi University, who made this technology, had to wait to get it. So many years the person has to. Deepak Pental, Delhi University, Deepak Pental, who created this GM mustard. This is only, mustard is essentially a self-pollinated crop. So what he did was that he just made some Barnes-Buster combination of genes borrowed from bacterium, and when one gene is present, the other gene will suppress it. Solely at the time of, pollen development. So in the case, what happens is that the pollen grain development is arrested in an essentially self-pollinated crop. Mustard cannot be improved genetically because you cannot cross it. It's self-pollinated. So only technology, and then it reverts back. But then you had to fight for 15 years to get this approved. But even now, it hasn't gone to the farmers' field, but only controlled trials are available. So Barnes-Buster, so it's very, you will get the idea of GM mustard if you look at it. But then still, though he had spent his lifetime on this, it's not being lavishly gone into the golden rice. Again, maybe that is a fancy kind of thing that you could get your vitamin A deficiency using this. It also has, again, a great genetic engineering program that is being done, and it was perfectly done, and I'm sure, especially in poor countries, African countries, they are benefited, because malnutrition, vitamin A deficiency causes night blindness in people. So once you have this, once you, don't look at me as former dean, you as a chairman. Okay, this is again BT, the golden rice, where the beta carotene is expressed, and iron fortified, anemic, So once you eat, so protein enriched, iron fortified, beta carotene enriched rice, if it comes in, what a kind of blessing it is. But how would you do that? How would you overcome that, the kind of mudslinging that's being done, and do it, that's to be seen. This is a creation of it. It's a very beautiful kind of thing. 3 genes borrowed from 2 from bacteria, and one from bacteria, and 2 from daffodil. And, it's not an easy thing. You are performing an experimentation on a live organism. And if you can precisely control it, don't you think that it's a wonderful science? Don't use golden rice, but accept this technology that we are in a position to manipulate the way we want. Beta carotene, if it is expressed, why do you bother about what is happening? There's not doing any harm to the people. Rice will not have beta carotene. So if you have rice being developed with that, why the people are, I'm sure that there are attorneys who will go to defend cases, and the judges who do not know much of it, they say that, okay, no, we will not allow this. It should not happen. The technology, science is so powerful. Science is for everyone. Science is for the society. Don't do anything, abuse science. When you abuse science, I'm sure that it's going to have many problems. But then if it is being used, and a scientist, I'm sure that he will not abuse. You will not become a Hitler or a person who would kill people like that, you know. So you would always look at the comforts of the other organisms and then you would work it. But then still, people don't have faith in. Maybe that somebody might have done something bad, but that doesn't mean that every time everyone should be branded. The science is the only truth that comes out from it. Science can only bring truth, isn't it? Understand? Science cannot be generated that way. It is through experimentation that you generate results, isn't it? So that's the truth. We accept it. All these value additions, all those great things that you have is mainly because of that. I'll just microbiome research, again, it's a very important area that you would, the gut microbiome, you know that they say that the microbiomes in your body is more than the number of cells that makes your body. You know, so that many. You have microbiomes in the gut microbiome, you would have microbiomes in the liver, and they do a wonderful job for you. Wonderful job for you. The gut microbiome is the one which helps you to digest your food. It stimulates your taste buds and probably make your liver cells to function properly. So microbiome, and that's why people say that the probiotic process, you know, microbiome enriched food is going to, nutraceuticals, which is enriched. And I'm sure that this is going to be a great science. Biodegradable plastics and environment. So again, you know, like even Dr. Ananda Ramakrishnan was talking about the wraps of your chocolates, which remains there like as such, you know. Maybe that the printing would go, but the plastic would remain there. So what will happen? There is no organisms. There are few organisms which could make degradation, but then I'm sure many of them, except that the one which was devised by Ananto Chakravarti, the Pseudomonas, which eats away hydrocarbon. So that would be something, if you refine it, Ananto Chakravarti, an Indian, made the first transgenic bacterium, Pseudomonas, which degrades, oil. So that again, otherwise, you know, you don't have anything. But you have alternate models. Alternate models in that you have Ralstonia, a Ralstonia species of bacterium, which produces polyhydroxybutyrate. Polyhydroxide is a polymer that is being released by this bacterium, and that becomes a biodegradable plastic. You don't call it as a biodegradable material, or what you call as a biomaterial. People are trying to use that in the big way. Now, coming to my last part, I'll just some of this, and I'm sure that you would get from the website. There's so much of opportunities available. National Postdoctoral Fellowship is being announced. You know, 80,000 rupees. You have a PhD and you get it for 2 years, 80,000 rupees. And you have just submitted your PhD, you will get 50,000 rupees, 2 lakh rupees per year for your contingency. What a blessed kind of thing. I didn't have that kind of, I started my career with 100 rupees fellowship, you know. But now you're getting 80,000 rupees if you are a postdoctoral fellow. National PDF, you know, which is being made by enough. These are all some of those DBT supporter systems. Again, a great opportunity for all of you. Bio E3 policy, biotechnology for employment, economy, and environment. So much of things are included in that. Millions of rupees are being put into this. And anyone, you can start a bio foundry, a common facility anywhere you want. You can go for bio manufacturing. You know, materials. You can go for just funds available. No questions asked. Somebody was telling, you know, that Ananda Ramakrishnan was telling that the money invested, he didn't get the return. You know, see, that's what is called as high risk, high value products. You know, high risk. It's risk in the sense that there will be a lot of risk in doing something, but then the value, if you succeed in one, I'm sure that government says that if 9 of them fail, and if one works, and that's great things, that's worth it. So maybe that there are a lot of bio E3 policy. It's called as circular economy, that you always go for circulate. You know, the economic and Bio E3 policy along with the bioeconomic policy that is being enacted by the government. It is released, where biological wealth, marine wealth, terrestrial wealth, microbial wealth, all these being blended together to either go for a biomanufacturing process, fermentation process. So many bioproducts can be produced this. And the government of India realizes it, that anything can be made from the biological systems. And so I'm sure that you all will feel proud to be associated with the Bio E3 policy. Try to. Just browse it. You'll get all the ideas what exactly is. It's a kind of Pandora's box that comes to you, that you open it and you would see the kind of benefits here. I'm not promoting Government of India's policies, but then Bio E3 is a magic word, which would, not only that, it's not, employment is also being safeguarded over there. So it is, that's what E3 is being put together. So bio-based chemicals, functional foods, precision biotherapeutics, precision medicine. Of course, genomics has gone for making personalized and precision medicines. So genomic understanding, precision medicine and personalized medicines are available, and they are all great things. So, but done with this concept, I'm sure that this being boosted, that you would be able to go in for making several things by using this. So public-private partnerships, that's what is lacking here. So once we do it, and I'm sure that the registrar is sitting here, we need to have something by which the private industries are to be brought in. That's what I've been telling them, because great things can happen because government investments may diminish down, so we need to have private. And I was also talking about let some industry come in, have their establishment here in the campus. They will establish facilities for you, and only thing is that you'll have to work as aligned to the industries. I'm sure the wonders will happen, and start incubation system, because startup culture, that's what I'm saying. Rather than becoming a job seeker, you can become a job provider by just doing a startup. Startup ecosystem is great in the country. BIRAC of the biotechnology, DBT, Department of Biotechnology, and the startup mission of Kerala government. They have supported several startups, so no questions asked. You will be, if you have an idea, if you have a good proof of concept for something, start an industry, start a startup, Eventually, like Biocon, Kiran Mazumdar, that you would become a millionaire within the course of time. And I'm sure that you would be able to employ all these people in your class into the establishment that way. So think about that. Think out of box. Think you have the great potential with you. Make use of that potential in you, and then try to go in for making things. And I'm sure that you would be a game changer in the course of time. So don't you want to make a challenge to yourself that I want to become a game changer? I want to establish a startup. Ideas would come, and then you consult people. There are mentors available. You talk to them, and then how to refine something by which you will be able to go. So I'll not. If you go into ANRF is the one 2023 government has enacted. SERB, Science Engineering Research Board, is now converted into ANRF. Dr. Deshmukh, who is joining here, is an ARF Prime Minister Professor. It's a new concept. You are retired, but then irrespective of the age, you want to come to a university from a great institution. The wisdom that you have, make use of it. And I'm sure you're lucky that Professor Deshmukh is joining in March as a Prime Minister. So these are all coming through the ANRF system. So just look into the ANRF website, and all this, you will get it over there. Young Scientist Program. So much of things are national PDF, partnership. As I said, that hub and spoke model of partnership. Then Vice Kiran for ladies, methane monitoring, these are all. Then these are some of those 2026 calls, so you just see that. J.C. Bose Grant, J.C. Bose Fellowship, anyone who is in the service can avail it. National Postdoctoral Fellowship, 17 February, 15 January it opens, and 17 February up to that you can apply if you are a postdoctoral fellow. Advanced research and ARG is something which goes into crores, there are National Science Chair, NSC, Ramanujan Fellowship, Ramalisinga Fellowship. See, you go abroad, absolutely no problem. You should go abroad. Go abroad and come back. Go abroad, and then we have this fellowship available. when, when he came, that was a clear indication that I should stop it. When he came, that was a kind of blunt. So I just very sorry. Okay, there is no elevator to success. You have to take the stairs. Now, maybe that if you want to become a startup, do a startup or do things, you should. I'll just end up. I'm just a plant biotechnologist. I didn't know anything about viruses. I didn't know about virology, but I learned virology to establish something like Institute of Advanced Virology when I was there. I learned virology. Took 2 years to learn rigorously, bring all the people from different parts of the country. If you see it, it was inaugurated in 2021. See that all these people, these are all big people from Christian Brasheau, Robert Gallo, all those big people, Sham Sundar, all these people were gathered because I didn't know anything. Government wanted to have a virology institute in the country. And maybe that with great difficulty, we gathered all the people, made a, DPR, and then, see all those big people they have given. They came for the inauguration. Just understand, a small institute, state institute to be inaugurated in Kerala. All these people flew from all the parts of the world to reach here to witness that. They said that they never think of such a big facility being created. And this is the group that we have, Institute of Advanced Virology. It's a great blessing for you. We had only one virology institute in the country, the National Institute of Virology, Pune. Now we have much better institute in the state of Kerala. And once you learn virology, because there are a lot of opportunities available. Dr. Rajendra Pilangata was, so he's there, no? He's there, okay. So all these people had helped me in establishing this. The institute is like this, added this, and I'm sure that if you pass through Kollam, Attingal, after Attingal, if you look at your right side, you would see the big building, 1 lakh square feet and flourishing institute. I feel so proud that I was part of, I could do something to establish this institute. So thank you very much, and you can do wonders. You know, that's what the multidisciplinary concept that you have, interdisciplinary concept you have. You don't have to be an expert by birth. You can acquire any expertise in the course of that. I don't say that I have done wonders because the government wanted to do it and came to me as I was advisor to the Kerala Biotechnology. Thank you, Chairs, taking all the time. How many? I'm so sorry.
Thank you, Professor. Sir, your talk on research ecosystem really inspiring and so much knowledgeable. You covered almost all the topics, and sir emphasized about the understanding the science in natural phenomena and how we can translate into innovation, and how we can go for scaling up. As we all know that, see, Carey Mullis, when he went for taking part in the hot spring waterfalls, he felt, why can't we isolate the bacteria, Thermophilus aquaticus? From that, we isolated enzyme for PCR, polymerase chain reaction. So, it's the same way you see C.V. Raman, when he was coming after his conference from London, over the sea, he observed the colors, various colors in the sea, in different colors. And he realized and studied this deep science, basic science in it. And he discovered that Raman effect. As we know that value of the Raman effect now. So, sir emphasized in his talk all the natural phenomena we have to observe and understand the basic science. That you can go for innovation. Sir, your talk is highly inspiring, and you covered almost all the area, especially how funding facilities are available for research scholars and faculty members in various funding agencies in India. How can, generally I can tell that ours, especially in biological science, so students, scholars will find out, for example, isolating one compound from one plant. Thereafter, that student will submit this, he will left. And the other student will come, they will try another plant and isolating the same compound. Instead of going on further application of that compound or scaling up or increasing the synthesizing compound. So such a research are emphasizing to take up by the student. I hope that your talk is highly inspiring and informative for us. With this, we thank wholeheartedly for your coming and given this talk. And sir is, we all know that sir is one of the academically achieved and contributed to our university and guiding our university, and especially we all know that the Department of Plant Science is established by Professor G.M. Nair sir. So thank you very much for coming over here and giving the talk in plenary talk in this first session related to plants and biological science. Thank you very much, sir. Thank you very much. I request our sir, please, sir. Please. No, we have session. Yes, yes.
I welcome Chair Professor K. Arun Kumar sir for handing over memento to Professor G.M.
I
Nair,
request
sir,
our head of the
as a
department, Professor Dennis Thomas, jointly with us to give memento to our professor. Please come.